me know. What we have done last time was we looked at animal research, right, beta? Specifically in terms of what was required in animal studies perspective. That's what was done last time. So um, again, I'm gonna share the screen with you so that we can, see the elaborate details of it, of where we are. So we had done animal ethical issues in animal studies. That was the sixth number, 1.4.6 version, which was completed. Next was compliance to ethical guidelines and dealing with ethical issues in terms of compensation and subject rights, which will be coming under the subparts where we were studying about NCE to clinical trials and phases. So that's the way how it goes into. So I'll just show you the screen with that part, and then we will straight away go ahead with the details. So for a second, I'm just sharing the screen first with the syllabi, just so that we are on the same page, guys. So you can see the syllabi being there. So in the syllabus, NC and its development in new drug, we did 1.4.6 ethical issues in animal studies. That was what we had done last. Seventh and eighth, compliance to ethical guidelines and dealing with ethical issues, subject compensation, subject rights, comes as 1.3 sections because this is all about clinical trials perspective. Okay, so we are going to go to these details now. Fine, guys. Do we have any questions on this, beta, or is it clear, or is it understandable?
Yes, sir, it's clear.
Okay, thank you, beta. Thanks for confirming. Thank you so much. So now what I'll do is I'll just open up the part which is required to be understood in that part. We had gone through drug targets and drug development, high throughput screening, okay, the methodologies which were being done from NCE point of view. So we need to now understand how a drug becomes a drug in a quick version, and then we will go down to the details of going ahead and understanding something called as the basics of clinical trials and introduction to clinical research. So doing that bit, before we go into that, first we will just how a drug becomes a drug, a quick one is what I intend to show you. So I'm just sharing the screen with you. Let's see, as soon as you can able to see it, and we will discuss it out, and then we go further from there. It's still showing sharing beta.
Drug research and development.
Can you see the screen, guys?
No, sir.
Okay. Okay, I'll share it again, beta. Can you see the screen, guys, now? Because it's showing presenting in my screen. It's a container with the tablets coming out. That's what is visible on the screen. Are you able to see? Hello?
Yes, sir.
Okay, madam. Thank you so much. So we are quickly looking at how a drug becomes a drug. Okay, it's just a 5-minute one, quickly glancing through, and then we'll go to the elaboration. Fine.
Aims to prevent or treat disease. While the end result, a small pill or capsule, seems so simple, the process for developing a safe and effective new drug is anything but. Development can take as long as 20 years and cost more than $1 billion.
So, but at 20 years, the mentioning is because the patenting period is for 20 years. So that's why we take it as a benchmarking standard as 20 years. The cost is of, as of today, $1 billion. That's the cost for developing a single drug. Okay, not many drugs, but single drug for a single entity.
A relay race, drug development has several stages and requires a team effort, often involving the government, universities, and pharmaceutical companies to reach the finish line. The process begins with basic research. U.S. government agencies, such as the National Institutes of Health, conduct and fund research at laboratories around the world to uncover fundamental knowledge about diseases.
So, beta, there is some organization. So you have to understand that drug research, okay, is not only by the companies. It also has entailments by companies, research centers, other organizations. All of this is being there in sync. So when we talk about drug research, it is primarily being supported by the big organizations like National Institute of Health and National Library for Medicine, Laboratory for Medicines, Library for Medicine, NLM, NIH groups are the ones which would be supporting it in a big term, because until unless basic academic research comes in the forefront, we do not have molecular leads. That means basic research जो होता है बहुत ज़रूरी होता है, उसके बेसिस पे ही आपका ड्रग कंपनीज आगे का कंटेक्स्ट करवा सकता है, क्योंकि बेसिक रिसर्च के परव्यू के बिना वी कांट गो अहेड एंड डू समथिंग कॉल्ड एज clinical trials, drug research or further. Okay, so NIH and NLM gives or supports basic research by providing the funding for it, and at the same time taking a tag on how the research is going on.
This research helps identify potential drug targets, usually genes or proteins whose functions are critical to the survival or spread of a disease-causing organism. Scientists then investigate how they can interfere with these targets to either control or eliminate disease. They may test tens of thousands of chemical or biological compounds to see if they either inhibit or stimulate a given target. Usually, only a very small percentage of these compounds will have an effect on the target. These hits are then rescreened multiple times to confirm the results and further trim the list of potential drug candidates. Researchers look.
So, beta, this method is what is called as high throughput screening method. We are trying to identify the targets. They are trying to figure out whether it is showing the relevant effects or not. So that's what is the first phenomenal part, which continues out, which is the one which is being done. So that is what happens in the first one. Okay. Next, after this is over, is the one when we go ahead and do the further part of it. So while high throughput screening also entails something called as QSAR, quantum structure activity relationship, it accelerates the overall process of identifying the targets, knowing the mode of action, and ascertaining that it is being easily understood in a faster technique or faster way.
For compounds that interact only with the desired target. If a compound reacts to unrelated targets, there is a greater chance of adverse side effects. To further minimize this risk, researchers conduct experiments to optimize a compound's absorption, distribution, and metabolism inside the body. These studies help determine which compounds are safe and effective enough for further testing. With the results from these preclinical studies, drug developers seek permission from the U.S. Food and Drug Administration to begin testing the compounds in people. If granted, scientists embark on a 3-phase clinical testing process that will determine whether a drug will be approved for public use. Because clinical testing requires a substantial investment, it is common for pharmaceutical companies to play a larger role during this stage. Phase 1 clinical trials test an experimental drug in about 20 to 80 healthy adults to evaluate.
So this is your phase 1 clinical trial, beta. So in a phase 1 clinical trial, you, except for 2 therapeutic areas, for all other therapeutic areas, you have something called as healthy volunteers. So you can see the green notion is that they do not have any other disease or any manifestation of it. So they are healthy. So why is it the healthy volunteers are tested? Because we do not know anything about the drug. So what happens is, if the drug is given to already immunocompromised person in the sense, a person whose immunity is already down. So what will happen is, that person would be more susceptible to an effect by the drug as compared to by any other compound. So to ensure that the drugs are not detrimental to the human body in any way, uh, is the first one that's called safety studies. So the safety studies would entail the use of healthy volunteers, and here the healthy volunteers are tested or are given the drugs and studied for a certain time duration, and that gives us the first informational idea about how the drug is performing in human subjects or human beings. Because prior to this, all the studies have been on animal studies or on in vitro regions where the human biosystems, that biological systems which we have, immune system, our nervous system, our overall homeostatic systems, which have been instrumental in working towards to keep
Hello, sir. Sir, we can't hear you.
Uh, Hi guys, welcome back. I don't know, again, I dropped off the call. I do not know exactly what happened. So, were you able to see the video and were you able to listen to what I was describing, beta, or should I be repeating it?
Yes, sir, we had listened, but
Oh,
in between it got cut.
okay, beta, fine, beta. I thought so because I did not know where exactly it was. Okay, fine. All right. Wait a sec, guys. Just Okay, guys, did you see till here, beta? This was a phase one clinical trial starting up, green colored one on the screen.
Yes, sir. Yes, sir.
Okay, okay. So, beta, I was explaining here that the phase one clinical trial is conducted on healthy subjects because your diseased person are immunocompromised. Immunocompromised means their immunity is already down, so we do not know how the drug molecular interface would be affected when it enters the body system. So the body system can have a detrimental effect of the drug. So hence we want to try first on healthy volunteers, and based on healthy volunteers, if everything is fine, then only we'll go ahead and give it to the patients. So that is what is the main onus of phase one clinical trials. However, there are two types of drugs or two therapeutic areas which can't be done using healthy volunteers. Number one is neuropsychiatric. So psychiatric drugs can't be given to healthy volunteers because that can turn them into psychological cases, which will be a little difficult to handle. Second is cancer drugs. Again, cancer drugs are supposed to work against anti-cancer or the cancerous lesions or cancerous cells in the body. So if you give it to a normal person, he may have hair fall and may have tendencies where some of the oncogenes may be triggered since there is no specific target site for the drug to act upon. Hence, in oncology and in neuropsychiatry, these are the two therapeutic areas where we do not conduct phase one clinical trial with only healthy volunteers. We conduct it only on patients and stuff further. All right, beta. So hope that is clear to you. Now playing further.
Yes, sir.
Thank you, beta. Thanks so
Yes,
much
sir.
for responding.
Evaluated safety, determine a safe dosage range, and identify side effects. In phase 2, the drug is given to approximately 100 to 300 people, including those with a target disease, to get an early indication of how the drug is working and to further evaluate its safety. Phase 3 trials evaluate the drug in a group of 1,000 to 3,000 people who have the disease. These trials
Hello,
So you could
sir.
see, guys.
No,
Huh,
sir.
beta?
Video is not playing properly. I'm just listening the audio.
Actually, you're not able to see the screen only. Okay.
No,
One
we
second.
can see, but it is not moving properly.
All right, beta. One sec. I'll just play it from a little ahead. Hmm, okay. Okay. Can you see the screen now, guys? University turn down.
Yes, sir.
Okay, I'll play it out. Just see if you can see now.
clinical studies, drug developers seek permission from the U.S. Food and Drug Administration to begin testing the compounds in people. If granted, scientists
Are you able to see it now, beta?
So it is in one position only, it's not moving.
Okay, beta. Then beta, the connectivity problem is at your end, beta. Because just see, beta. Just एक बार check
Maybe.
कर लो. Because here I'm having the second system which I'm playing so that I can be sure that you are able to see. One second, I'll just play it again now.
embark on a three-phase
Okay,
clinical testing process
sir.
that will determine whether a drug will be approved for public use. Because clinical testing requires a substantial investment, it is common for pharmaceutical companies to play a larger role during this stage. Phase 1 clinical trials test an experimental drug in about 20 to 80 healthy adults to evaluate its safety, determine a safe dosage range, and identify side effects. In Phase 2, the drug is given to approximately 100 to 300 people, including those with the target disease, to get an early indication of how the drug is working and to further evaluate its safety. Phase 3 trials evaluate the drug in a group of 1,000 to 3,000 people who have the disease. These trials aim to confirm the drug's effectiveness and monitor side effects. They also may compare the drug to commonly used treatments or to no treatment at all. The larger size of Phase 3 trials allows for results that are more statistically significant or less likely to have occurred by chance. If a drug successfully completes Phase 3 testing, a company will seek permission from the FDA to market the compound. Its application contains data from all the preclinical studies and clinical trials, along with information that the FDA will need to make its decision on the drug's safety, efficacy, and quality. If approved, the drug can be manufactured and sold to prevent or treat the disease in question. But the process doesn't stop there. The FDA will continue to monitor the drug's safety and effectiveness for as long as it remains on the market.
So beta, this is what is about the picture which you get to know from new chemical entity to prodrug to approvals and finally becoming a drug. Okay. Was the video clear, beta?
Yes, sir.
Yes, sir.
Okay, thank you, beta. Thanks so much for confirming, beta. It's good that you guys inform, so that's nice. Okay, so next what we have is, once we know about this part, we are supposed to understand something about introduction of clinical research now to you. Okay, so I'm going to be doing that part now, introducing you to the subject of clinical research, where we will understand basic clinical trials in detail. So playing that by showing you a video about it.
Hey guys, thanks for watching this presentation, Introduction to Clinical Research for the Clinical Trials Guru. One of the things I get asked the most by far and have been asked to create is some kind of just...
Hello, beta. Can you see the screen?
Yes, sir.
Yes, sir.
Okay, just one confirmation, beta. Have I shown this slide decks to you earlier?
No, sir.
No, sir.
Yeah, I think so, beta. Are you sure you haven't seen or you have seen? Okay.
No, sir.
No, sir, we haven't seen this.
You must have seen it, beta, because that's what I am remembering. Just one sec, beta. So I'm just moving to the next one which you have not seen.
So one announcement before we get started. So
this is the one which you are not seen, beta. This is the basics of clinical trials. Okay, which is a long one, long version one. Okay, which is about details of clinical trials, which I would be using George Howard's slide deck presentation for explaining the concepts to you. Okay, playing it for you now, guys. Okay.
So the faculty software interface failed over the weekend, and so I haven't gotten you your information about your teams and your information for how to access the questions for your review of the material from the weeks before. Hope I'll have that done by the end of the day today or by tomorrow at the latest. And if the conventional software interface I can't master right away, we'll figure out a way to work around that. So I'm sorry for the delay, and I'm sorry I didn't communicate about that. It was the out of town over the weekend syndrome. So it's terrific to have today Dr. George Howard as our speaker. He's professor in the Department of Biostatistics here at UAB. Just a real bit of brief history. So he did most of his training and early career at the University of North Carolina. Came to UAB, I think, in 1999 as department chair for biostatistics and served in that role till around 2011. And as you said, he has the best job in the world now, ex-department chair, which I can relate to. His research has been wide-ranging, but really probably the highest impact and still very, very active is that he was the originating PI for the regards study. I can never remember exactly what regards stands for.
Reasons for geographic and racial differences in stroke.
Right.
You can't do research if you can't do acronyms.
Right. And this is a huge national study that has, I don't know how many times it's been renewed, must be 3 or 4 cycles now. It has an incredible amount of data that's been used well beyond the stroke aspect of the original intent of the study. And Dr. Howard stays as the a major user of the data, but also as a national coordinator for access to that information. You know, I think if you have any interest in accessing information from large countrywide research databases, he's a terrific person to talk to about how to make that happen most smoothly and what issues you have to confront, how you deal with the IRB issues, how you deal with all of the stuff associated with accessing data on a big trial. He's also the director of a national database on it's called CRESS, which is has to do with revascularization of carotid arteries for stroke prevention, and is just a great resource in biostatistics. And he's going to talk today about phase 3 clinical trials. And
Thank you.
If he stands here, we don't have our lapel mic today, so if he stands here, he'll be easily heard by the people online, and I'll have this microphone to walk around with. The people who are joining us online can hear faintly if you talk in a normal voice without the microphone, but if you really speak strongly with a normal voice, the microphones in the ceiling work pretty well. So if you have a comment, just please speak up. I may need to give this to Dr. Howard because he likes to roam. And,
No, I'm honored to be here. Please, I can't stand lectures, and please consider this a discussion more than a lecture. Please feel invited to stop me at any time. There are a ton of slides, and if we don't make it to the end of the slide deck, I consider that good, not bad, because that means we've discussed and gone into more depth on other things. And so please absolutely feel invited to stop me at any time. I'll also say that this lecture is a mess. Basically, this is a semester-long course in the School of Public Health taught in one day here. And so you will have the feeling that it is wandering through 1,000 different topics and jumping from topic to topic. And the reason for that is we're going to wander through 1,000 different topics and jump from topic to topic. And so basically, if you seem confused or something, just slow me down. Slow me down and we'll figure this out together. Basically, there's a lot to cover in something as simple as clinical trials. They really are simple. That's one of the beautiful things about them. And so basically, I'd like to put this in a little bit of context. And so basically, if you consider what is the spectrum of evidence, why do we believe what we believe? And basically, studies can be sort of ranked as from the weakest to the strongest, and clearly the weakest are what are called ecologic studies. And ecologic studies are that people in the south of France drink a lot of wine, and people in the south of France tend not to die of heart disease. Gee, I bet it's because they drink a lot of wine in southern France. And the key of an ecologic study is I didn't measure anything on anybody. I measured outcomes for a group of people and exposures for a group of people, and then I guess that they are associated. Sort of moving up the food chain, the next level is observational epidemiology, something dear to my heart. And within that, there's also sort of a ranking, a food chain of studies. And I wish I had more time to talk about all of these, and I do hope they're covered in your experience. But sort of the weakest is a case control, where I get sick people and well people and look backwards to see what made them sick and well. The next level up is a cross-sectional design. You can think of that being a snapshot. I take a picture at one point in time and see who's sick and who's well and who's exposed and who's not exposed. There's no temporality in a case control study. The highest on the food chain of observational studies is the prospective cohort. And basically the idea here is I get a group of people who generally don't have the disease, and I measure a whole bunch of stuff, and then I wait patiently, sort of vulture-like, to see who develops disease and who doesn't get the disease, and then say, aha, it was the people who smoked who got lung cancer later. And so that's it. And then finally, is this what we're talking about today, the randomized clinical trial? It's always referred to as the gold standard evidence. And it's amazing how few people don't really understand why it's the gold standard evidence. Wanted to take a second and make sure you're cool with that. And so basically, in the 1990s, I'm old. In the 1990s, there were a boatload of beautifully done studies that clearly demonstrated hormone replacement was protective of heart disease and stroke in women. In fact, I stood at the podium of the Heart Epi meeting and put my arm around Trudy Bush, God rest her soul, and said, we can never mount the Women's Health Initiative because we all know for sure that hormone replacement protects women from heart disease. It is unethical to do this study. And then 3 clinical trials with many fewer patients in the observational studies suggested hormone replacement was not only protective, in fact, it was harmful. You know, the entire field just dumped those epi studies and went with the clinical trials. It's now dogma that hormone replacement's not cool. You know, how can that occur? Why do we accept the results of the RCTs over the results of those incredibly well done, beautifully done observational epi studies? And so the idea of this is an important topic for today, which is confounding. I want to make sure, by the way, at different points in my lectures, I always say, this is an important thing. I'd like you to remember that. I'll say a bunch of stuff that must not be important because I don't say it then, but this is one of the things I'd like you to take home. This is one of the things so basically a confounder is I have some exposure, estrogen, and I'm interested in seeing if it's related to some outcome, heart disease. Okay, so that's the relationship I'm interested in. Okay, but I've got this other thing hanging around in the ether called a confounder, and it's not, but this is probably socioeconomic status. Probably socioeconomic status. And it turns out that rich white women took hormone replacement. I mean, you know, rich white women don't have heart disease. Okay, and so instead, what we were fooled into thinking was that by the way, by the way, people define things differently in science. I'm sure you've noticed that. My definition of a confounder is anything that's associated with both the risk factor and the outcome. Okay, if you're a hardcore epidemiologist, there's a little phrase that is added that says, and it's not in the pathway. Okay, I'm not smart enough to know what's in the pathway and what's not in the pathway. And my example of that is always inflammation. You know, inflammation is causative probably of atherosclerosis. And by the way, atherosclerosis is an inflammatory process that increases inflammation, so the things are pointing at each other. Is it in the pathway or not? I don't have a clue. My definition is anything that's associated with both the risk factor and the outcome. And these can lead to false associations, and that's what happened here. Okay, and so in this case, rich white women are more likely to take hormone replacement, and rich white women are less likely to have heart disease. So it looks like hormone replacement protected you from heart disease. Okay, so how does randomization fix confounding? Well, with randomization, the high SES women and the low SES women were equally exposed to the risk factor. Okay, and so basically that leg of the three-legged table is broken. Okay? And it doesn't matter that the confounder is still related to the outcome, because things cannot be a confounder unless they're related to both the exposure and the outcome. And randomization breaks that three-legged table, and that's cool. That's cool. And remember this, that's the cool thing about randomization. Now, randomization is wonderful for many reasons. I wish I could talk more about it. But the single biggest reason is not only are high and low SES women equally exposed, but there's equal exposure for all risk factors. For all risk factors, even the ones we don't know about. It fixes the confounders for factors that you're not even aware of yet. It turns out observational epi, and I think I say this here. So I don't know if I say this, I might say this in a minute. I've forgotten what I'm doing next. But anyway, so the cool thing about randomization is that it does it for both the ones you know about and the ones you don't know about. And so why not do all randomized trial? And the reason for that is research is a process. You start out with ecologic studies, you move through randomization, and then finally at the end of the day, when it's really well understood, you do the trial to put nails in the coffin. You don't start by doing the randomized clinical trial. They're really expensive. The CREST trial that was mentioned in the introduction is a $65 million study, and you don't frivolously do $69 million studies. And basically, they only address one question at a time. So if you think about it, that's a boatload of money to answer one question. And also, some things just can't be done with randomized clinical trial. We all believe seatbelts prevent deaths in cars. There's never been a randomized trial of seatbelts. Interestingly, the tobacco companies, I used to be from Winston-Salem, a town, the cigarettes were named for the town, not the town for the cigarettes, by the way. But anyway, and the tobacco companies are so cool. They come out and they say, there has never been a definitive evidence that cigarettes cause lung cancer. And I agree 100% with them. Absolutely a true statement, okay? To do that, you would need to randomize children to smoke and not smoke, and then follow them forward and wait for who develops lung cancer. And then we would have the proof that cigarettes cause lung cancer. And the people who buy into this, I don't believe it if it's not a clinical trial, should be comfortable with the tobacco company's correct claim, that there's no definitive evidence of a link, because they're right, okay? But anyway, so a lot of things can't be done. Parachutes, never been a randomized trial of parachutes. It's just not going to get done. Okay, so in terms of the big picture of trials, and all this is just leading in, I'm talking about phase 3 trials, and it's important to put those in the context of the clinical trials process. And so the first trial is called a phase 1 trial. By the way, this phase 1, 2, 3, 4, different definitions by different people, so watch out. These are my definitions, and most people agree with me. Other people have different definitions. A phase 1 trial is simply, is the drug tolerable? The first in humans attempts. If I give this to somebody, do they die on the spot? Typically done, particularly in cancer trials, in very sick people, just to make sure the drug is cool. Okay, the next is where we begin to get different definitions. A phase 2 trial, early efficacy trials. And they're usually divided into what's called phase 2A and 2B. A phase 2A trial is a safety dosing study, usually giving the drug and looking at the levels of the drug in the serum. Are we delivering? Because if you think about it, where we're doing is moving from giving drugs to rats to giving drugs to people. And if you start trying to guess, well, rat's about that big, and I use this much drug, and the human's about this big, we're just going to multiply the height of the rat by the height of the person, which is pretty much what you do to get the dosing for the person. Okay, might guess wrong. So the first level is safety dosing. The second is early efficacy assessments. That's phase 2B. Okay, early efficacy assessments look at surrogate outcomes. Okay, so I'm not going to look to see if we prevent stroke. I'm going to look if we prevent stroke on a CT scan, okay, which is a subclinical stroke. So I'm going to look to see if it stops progression of carotid atherosclerosis. Because if a phase 2 trial had the same outcome as a phase 3 trial, it would be called a phase 3 trial, okay? Then finally, what we're talking about today is the pivotal trial. Does safety, efficacy, meets regulatory requirements? Importantly, it doesn't answer all the questions all the time. By the way, y'all should be asking questions too, so stop me. Remember, I wanted you to stop me. Anyway, so next is the post-marketing assessments, the, further safety trials. And so why don't we nail down safety in the phase 3? Well, the phase 3 trial is powered to go after efficacy. Does the thing work? It's not powered to go after safety. Is this thing safe to use? So the example of that, perfect example, is Vioxx. Okay, Vioxx, incredibly efficacious, stops pain, works great, okay, but it increases heart disease risk. But the heart disease risk is still so rare that the study's not big enough to see it in the phase 3 trial. And so we have to do this post-marketing assessments of safety afterwards. And so this is really all a process, but for today, we're just focusing on the phase 3 trial. Everybody cool? Yes, ma'am. Thank you.
In phase 4 trials, when you say post-marketing, is that like the FDA gives like, okay.
Guys, this is an explanation and a discussion going on, and we are getting to learn from here. So any questions here as of now, beta? Phase 1 is toleration, phase 2 is early efficacy, phase 3 is pivotal, and phase 4 is post-marketing assessment. This is the types of clinical trials. Any questions you have? Do you want me to clarify anything? Okay, fine. So it's clear. Good. Okay, so I'm playing it further, beta. Okay.
We start seeing commercials for it, and there's still trials going
Yeah,
on?
exactly. The question was, is this something basically required by the FDA? And so exactly, that's precisely what happens. The FDA approves the drug, but says, oh, we're a little bit worried about heart attacks with your Vioxx. And we're going to require you to collect data systematically to make sure that people aren't dropping dead while their knees feel great. Okay? And so basically, usually they're required by the FDA. FDA is incredibly, phenomenally powerful. Someday you will have an FDA audit and you will be impressed by how powerful your FDA is. It's an amazing process. Okay, so here is the outline for today. This is all we need to cover today. Okay, and this is why it's going to seem like we're hopping around a whole lot. Okay. And when I teach this as a course, each one of those brown things is a separate lecture. And so what we're going to do is 3 slides summarizing a whole bunch of a semester-long lectures. Okay? So don't get too confused. Try to bear with me. We're going to start off with the ethics of clinical trial. It's really hard. I mean, if you think about it, all a clinical trial is human experimentation. That's all it is. Okay? We're randomizing people to different treatments. The barrier between us and the Nazis is phenomenally small. I mean, it is phenomenal. I mean, stop and think. If you're a clinician, most of you, I assume, are clinicians. When you're asking somebody to be in a trial, that is a huge ask. I mean, that's not a small thing. And the key to it is equipoise. And a lot of people don't understand equipoise. I mean, are any of y'all surgeons in the group? Oh, I get to insult surgeons. They're online. Okay, I'm sorry. Please forgive me. I've never met a surgeon who's an equipoise. I mean, these guys and gals know with certainty that cutting you open is going to make you better. I mean, they don't think, and by the way, I want them to think that. I mean, that's not a bad thing. That's a good thing. If somebody is cutting me open, I want him or her to have a very deep belief that they've got a good reason to do it. Okay? Equipoise is not whether I doubt it, it's whether we doubt it. Whether, in your opinion, there's not a collective body of evidence that we share and agree on that this treatment is good. Equipoise is a community statement, not an individual statement. And you can remember that, again, because surgeons are never at equipoise. They're never at equipoise. Okay, again, clinical community versus, oh, by the way, I love how we do trials and how the Europe does trials. They have a different principle. They deal with what's called the uncertainty principle. And the uncertainty principle, if you do a European trial or you do trials with Europeans, they put people in studies when the individual PI is uncertain what study is best, what treatment is best. Okay, fundamentally different approach. You know, in the U.S., we get all over sites who don't put eligible people in studies because we worry about whether it's generalizable to the entire group. And so if we find out that you're not putting somebody in the study who's eligible, we come down on you like a ton of bricks. That's different in Europe. They say, oh, I wasn't sure. I was pretty sure, you know, this treatment would help the person. I'm not going to put them in. So they're, you know, that's not a subtle difference. Okay, and that brings up the whole topic of informed consent. And how many of y'all have consented patients for studies? So everybody has. Do you think they understood what you were saying? Tell the truth. I mean, I mean, here's the cynical comment for today. Informed consent is to protect the university. It is not to protect the patient. I mean, regards, which I was brought up at the beginning of this, the most invasive thing we do is draw blood. That's the worst thing we do to people. Our informed consent is 8 pages long. Can you believe? I mean, that's amazing. The worst thing we do is stick a needle in the arm. Anyway, and so so stop and think. I think we do have a duty to have people understand because what we're asking them to do is, as I said, a huge ask. But in general, that's going to take more than going over the informed consent document. I think it really will require a discussion with these people that goes quite beyond that. So the next topic, if you look at that outline, was the different type of hypotheses. Okay, and so basically, what I want to do is have a hypothesis that's clear, quantifiable, simple, singular, and doing one thing in a clinical trial. And what it does is give the who, what, and when of the study. This is the CREST primary hypothesis, and it's to contrast the relative efficacy of stenting and endarterectomy, that's the treatment, and preventing the stroke, myocardial death, and during a 30-day procedural period, and stroke afterwards. That's the what and the when. And then in patients with symptomatic or asymptomatic extracranial stenosis, that's the who. So when you're thinking about writing a hypothesis, make sure you've got the who, what, and when are all there. And it's amazing when you read these from even big trials, a lot of times, I mean, you should be able to give your hypothesis statement to somebody and have them pretty much understand what you're doing. I mean, they won't understand the details, but they'll get it the 30,000-foot view. Okay, now let's get into something that is more, a little bit more complex, which is, and people talk about superiority trials and inferiority or equivalence trials. And this is sort of an important thing and a different way of thinking. So I'd like to take a few minutes and just go over those and make sure that you're cool with this. And so basically the idea of a superiority trial is to address if 2 treatments are the same. So by the way, in all of statistical testing, whatever you want to prove is true, you assume is false. Okay, then you go out and collect data and try to prove how silly you were in saying that it was false. That's the whole idea of statistical testing. So if you're doing a superiority trial and I'm trying to prove 2 things are different, I assume that they're the same. I assume that whatever I'm trying to prove is not true. I conduct an experiment to prove how different they are. And, you know, basically what I have is, suppose there's a new treatment and an old treatment. And I want to show the new treatment is as good, really not worse than, the old treatment. That's a whole different way of thinking. That's a lot different than proving that they're the same. If I want to show that I'm setting out to prove they're as good. So let's say the new treatment is less invasive and costs less. Okay, so I don't need to prove that it's better. I only have to prove that it's as good. Because if I'll save money and I won't have to do surgery, for example. Yes, ma'am.
So this is why it confused me with drugs. If there is a new drug and the randomized trial at the end compares that drug to the standard of care, and there is no difference, but the drug, for whatever reason, will cost less, the FDA can then still approve it, even though there's no benefit to the patient.
This is a great question, and this is exactly what I'm talking about. This is what this, I tell you, let me get to the end of this. So the question was, if you do a trial and there's no difference, can the FDA still approve it and claim that it's as good? The answer to that is, it depends on what their hypothesis was. Okay, and that's what the difference that I'm trying to bring up here is. And so basically, and this is important, if you're doing a superiority trial, trying to prove two things are different, I assume that they're the same. And the trial, I hate this word, it's a negative trial. You don't see a difference. There are two reasons why it could be the same. Okay. The first reason is it's the same. Okay, it really is the same. And the second reason is I didn't have enough people in my study to see whatever difference was really there. Okay. There's no way on earth you can know which one of those is happening. Once you do a superiority trial and there's no difference, you can never know which one of those two things happen. It's impossible. Okay? So basically, you cannot tell which is true. One never, by the way, in all of statistical testing, this is one of those take-home points. One never accepts the null hypothesis. Okay, one only fails to reject it. Let me say that again because that one's important. In all of statistical testing, you never accept the null hypothesis. You would never say that they're the same. What you would say is that, by the way, this implies that traditional studies cannot be used to show that the new product is as good as the old. So if it comes out negative, what you say, and this is on the next slide, you end up with sort of a kiss your sister kind of statement. There's not sufficient evidence in these data to show that the two groups are different. Intentionally a double negative statement. Let me say that one again. There's not sufficient evidence in these data to show that the two groups are different. Okay, that's a whole lot different than saying they're the same.
Can I ask you a question about that last slide?
Sure.
So you said you can't tell the difference between if there's no difference or study size is the sample size isn't large enough. What if you have a very large sample size?
Well, it's still not large enough. By the way, there are no two things in nature that are the same.
So guys, this was about what exactly is the hypothesis versus, where you were comparing superiority versus equivalence, which is all the time studied, all this time looked out in clinical research in terms of looking at the best fitment for the drug to be taken further for studies and being released out for human usage. So the statistical assumption is the null hypothesis says that two treatments are same as compared to if there is an existing treatment, or conduct an experiment to prove that they are different. That's the way how it goes out. All right. So this is the superiority versus equivalence trial details which he has mentioned out, which is one of the most driving factor for checking out the goodness of the new product versus the old one. Is it clear, guys? Do you have any questions here? कुछ ऐसा बेटा जो आपको नहीं समझ में आया? हाँ बोलो बेटा.
Sir, what is randomization?
अच्छा बेटा, रैंडमाइजेशन का मतलब होता है बेटा कि सी बेटा आप अगर कोई सपोज आप डॉक्टर हो, ठीक है, और एक ट्रायल कंडक्ट कर रहे हो, ठीक है, तो आप आपके पास पेशेंट आएंगे और आप अगर चाहते हो कि वो ट्रायल सक्सेसफुल हो, तो आप क्या कर सकते हो कि जो सिलेक्टेड बंदे रहेंगे, पेशेंट्स रहेंगे, उसमें आपको मालूम रहेगा ना कि किसको कितना बीमारी है, कौन से स्टेज पे है. तो आप क्या कर सकते हो कि जो मतलब इनिशियल स्टेज पे है, जिसको अभी अभी जस्ट डिजीज हुआ है, जिसको थोड़े से मेडिकेशन से ट्रीटमेंट हो सकता है, आप उसे ट्रीट कर सकते हो और उसको आप ट्रीटमेंट ग्रुप में डाल दो। और जो लोग बाकी ऐसे नॉर्मल हैं, जो लोग मतलब बहुत ज़्यादा हाई लेवल पे, यानी दोस्त दो यार एकदम स्टेजेस विच आर नॉन ट्रीटेबल वाला पॉइंट ऑफ व्यू पहुंच गए हैं, उन्हें आप कंट्रोल ग्रुप में डाल दो और कंट्रोल ग्रुप में वो मेडिसिन दे दो जो उसके लिए ट्रीटमेंट के लिए यूज आउट होता है, और इस ग्रुप में, टेस्ट ग्रुप में, आप वो मेडिसिन दे दो जो आपका मेडिसिन है। तो ऐसा करेंगे तो क्या होगा, आपका मेडिसिन का इफेक्ट ज़्यादा अच्छा दिखेगा। डू यू एग्री? क्योंकि आपने जो ट्रीट किया है बेटा, वो लोग तो ज़्यादा सीरियस नहीं है। फाइन। तो अगर ऐसा आप करते हो तो क्या हो जाएगा, स्टडी बायस्ड हो जाएगा। डू यू एग्री बेटो? हेलो?
यस सर।
प्लीज रिस्पॉन्ड। ओके बेटा, तो अगर ऐसा होता है तो इसको बायस ट्रायल बोलेंगे। बायस मतलब जिसमें आपने जानबूझकर अपने इंटरवेंशन से स्टडी को आपके हिसाब से बना दिया है। तो अगर अनबायस्ड करना रहेगा तो आपको क्या करना पड़ेगा, रैंडमाइजेशन करना पड़ेगा। रैंडमाइजेशन मतलब यानी आपको मालूम नहीं होना चाहिए कि आप किसको मेडिसिन देने वाले हो। ओके, पेशेंट लिस्ट में किसका कितना इफेक्ट है। तो अगर आपको ना मालूम हो और आप रैंडमली मेडिसिन दो, उसको लाइक रैंडमाइजेशन, तो उसके लिए आप क्या करोगे, तो उसके लिए क्या करते हैं बेटा, पेशेंट्स के नाम के बजाय में पेशेंट्स को एक नंबर दे देते हैं, से ले 1 to 100। ओके, फिर आपको रैंडमली सेलेक्ट करने को बोला जाएगा। रैंडमली सेलेक्ट करने में तो हो सकता है, सबसे सिंपलेस्ट मेथड क्या रहेगा, 1 से लेकर 100 का एक चिट्स बनाओ। ओके, वो चिट्स आपके फ्रेंड को दे दो कि भाई इसमें से चलो 50 पिकअप करो। तो रैंडमली जो भी 50 नंबर वो पिकअप करेगा, वो नंबर के पेशेंट्स को आप एक को ट्रायल में डालोगे और एक को कंट्रोल में डालोगे। ओके, टेस्ट एंड कंट्रोल में डालोगे। तो क्या हो जाएगा, इसमें आपने कुछ बायस नहीं किया है। तो ये जो प्रोसीजर जो आपने किया, जिसमें रैंडमली लोगों को एलोकेट किया मेडिकेशंस और ट्रायल, उस प्रोसेस को हम लोग रैंडमाइजेशन बोलते हैं। इज इट क्लियर बेटा?
सर, तो इफ यू डोंट नो कि किसको कौन सा ड्रग दिया है या प्लेसिबो दिया है, तो हाउ विल बी डिटरमाइन आगे?
हाँ बेटा, तो बेटा आपको नहीं मालूम है क्योंकि आप, हाँ तो बता आपको मालूम है कि आप फिजिशियन हो इसके लिए, ठीक है। बट बेटा फार्मेसी वाला जो बंदा है, ओके, फार्मासिस्ट यानी जो मेडिसिन डिस्पेंस कर रहा है, ठीक है, उसको मालूम रहेगा कि किसको क्या मेडिसिन मिल रहा है। आपको समझ में आया बेटा? तो देयर इज समथिंग कॉल्ड एज ब्लाइंडिंग। ओके, तो हमेशा एक ट्रायल के लिए एक वर्ड यूज करते हैं, ब्लाइंडिंग। तो ब्लाइंडिंग मतलब पेशेंट को नहीं मालूम है कि उसको क्या मेडिसिन मिल रहा है और डॉक्टर को नहीं मालूम है कि वो क्या मेडिसिन दे रहा है। तो दैट इज कॉल्ड डबल ब्लाइंड। ओके, नाइदर द पेशेंट नोज नॉर द डॉक्टर नोज। बट ये दोनों को नहीं मालूम, इसका मतलब ये नहीं मालूम है कि हम लोग ट्रैक नहीं करेंगे कौन सा मेडिसिन जा रहा है। वो फार्मासिस्ट के पास है, फार्मासिस्ट नोज कि किसको क्या मेडिकेशन गया है। ओके, बट फार्मासिस्ट तो उस ट्रायल में दिखेगा नहीं ना, एक्चुअल पार्टिसिपेशन में फार्मासिस्ट नहीं है, वो तो डिस्पेंसिंग वाला, वो तो किससे करवा रहा है डिस्पेंसिंग, मेडिसिन डिस्पेंसिंग आपके डॉक्टर से करवा रहा है। मेक सेंस? सो द स्पॉन्सर, द फार्मासिस्ट नो इट बेटा, बट द एक्चुअल पेशेंट and the doctor don't know it, then that's called as double blind. मतलब they are blinded to the knowledge about what the medication needs. And फिर उसका जो रिजल्ट आएगा उस टाइम पे everyone will come down to know about it, क्या रिजल्ट हुआ। तो what happens is the entire trial is absolutely unbiased। इस प्रोसेस को हम लोग रैंडमाइजेशन बोलते हैं। फाइन, क्लियर नाउ? ओके, सो प्लेइंग फर्दर बेटा।
Okay, the intelligence of men and women is not the same. A radical statement, get me in trouble. I don't know which one's smarter, but the chance that two things are exactly precisely the same is impossible, statistically impossible. There is nobody 5 feet tall in the world. Okay, we just haven't measured people well enough. If we keep measuring them with sufficient precision, we will find out this person is either a smidgen above or a smidgen below 5 feet. There's nobody 5 feet tall. Okay, the philosophical part of this is that means the null hypothesis is always false. The two groups are always different. Okay, I just can't tell in what direction or how big it is. And so that all that is to answer if the sample size was big enough, I would have found the difference that I know exists with certainty. But that's sort of philosophical, statistical philosophy instead of anything. Okay, so basically, so the answer is, if I want to prove two things are the same or one is as good, what I have to do is to change the form of my hypothesis. I have to use a different form of my hypothesis. Okay, and so instead, and this was work by black welders, frequently also called black welder trials or superiority or non-inferiority trials. And so the new hypothesis is the new treatment is worse than the old. That is my null hypothesis. And my alternative hypothesis is now the new treatment is as good or better than my old. I've turned the hypothesis inside out. I want to leave this up here for a minute because I do see some wrinkled brows. And does everybody understand that what I'm doing is playing a game with the hypothesis? I'm changing to what I want to prove is true, I assume is false. If I want to prove it's as good, well, I'm going to assume that's not true. That means that the new treatment is worse than the old. And now I'm set up to prove it is as good or better than the old. Pretty cool, huh? Cool idea. Really backwards thinking, okay? And so if we reject the null hypothesis, then we can make a definitive statement that the new treatment is as good or better, because I can accept my alternative hypothesis in statistical testing. I just can't accept my null hypothesis. Everybody cool? Okay, so simple in concept, non-inferiority, sometimes referred to as equivalency, sometimes referred to as black welder trials. There is a slight difference between those. Don't worry about it. At the level you're at, just think of them being the same. Okay, and so then basically, regardless of the form of the hypothesis, a study provides an estimate of the difference between the treatments. I'm going to do a trial and estimate how different the two treatments are. I'm going to calculate 95% confidence bounds around those estimates. Here's a cool thing. There's a link between hypothesis testing and estimation. And basically, if whatever I hypothesize the difference is inside of the confidence limits, I will always fail to reject my null hypothesis. Alternatively, if whatever I'm assuming is outside of the 95% confidence limits, I'll always reject. And I'll show you what I mean by this. Right now, I see more crinkled brows here, but you'll get this in a minute. So basically, so what I'm going to do is a little bit of background. Let's say I have two treatments or products. I have an established treatment and a new treatment. I have a dichotomous outcome, a yes-no outcome. And I just want to calculate the percent of events in the established treatment. I'm going to call that pi sub 0. I don't do a lot of math. Trust me, don't panic if you're a math phone. Then the number of events in the alternative treatment, pi n, and in general, which treatment is best. So traditional hypothesis testing, superiority testing, traditional, okay? The null hypothesis, no difference between the treatment groups. So I'm going to say that pi 0 is equal to pi n. Okay, alternative hypothesis is the groups are different. Okay, that pi 0 is not equal to pi n. This is not hard, by the way. Even I can do this. This is easy. Okay, so basically, if you do that, then what I have here is a bar, and on this side of it, I'm saying the old treatment is better, and that side I'm saying the new treatment is better. So what I do is I calculate the difference in those two proportions. And I can put a little bar on my graph and say, there it is. Okay. And then I can calculate 95% confidence bounds around that. And draw those. Okay. Now, if that happens, if the 95% confidence limits do not include 0, then the two groups are different. That's how simple traditional testing is. So basically, if you think about it, there are three things that can happen if you do a traditional superiority trial. Okay. You can claim that the new treatment is better than the old. You can claim that there's no difference between the two treatments. or you can claim the old treatment is better. Okay, those are the two things. And by the way, I just fundamentally messed up. I said you can claim the two groups are the same. I should have said that was a slip and a bad one in this class. Okay, what should I have said? No evidence in these data that the two groups are different. And by the way, going back to your sample size question, basically, if you increase the sample size, you keep making those confidence bounds itsy-bitsier. And if I keep on making them itsy-bitsier, I will always find differences. We're going to come to how itsy-bitsy you want them as part of this lecture, by the way. Okay, so what are the advantages and disadvantages of a traditional study? The advantage is I can make a definitive statement that one treatment is better or worse. Okay. The disadvantage is if there's not an evidence of a difference, I cannot say that the two treatments are the same. I should not do that. Okay. The null hypothesis is not, by the way, in a lecture, when anybody gives a lecture and they say the same thing like 20 times, that's because they think it's really important. Okay. So the null hypothesis is never accepted. It's simply a case where you fail to reject it and you end up making a statement that's not sufficient evidence in these data to establish that a difference exists. Okay, and but the manufacturer sometimes only wants to say that the new treatment is as good. And if you do that, you do an equivalency analysis. Same picture. The goal here is to make a definitive statement that the new treatment is as good or better than the old. The null hypothesis is now the new treatment is worse than the old treatment. And here's the twist, plus a fudge factor. Okay, plus a fudge factor. And so basically what you say is that pi sub 0 plus the fudge factor is less than the new treatment. Okay. And the alternative is that the new treatment is as good or better than the old treatment plus the cushion. Okay, which is the pi 0 plus the fudge factor is bigger than delta. Yeah. Okay, and so basically, therefore, the definition is as good really means that it's within the cushion. Every superiority trial you will ever read has a fudge factor. When you're reading these, you should be, by the way, in the discussion, if it comes out as a positive inferiority study, they say the new treatment is as good or better than the old. What they really should say is the new treatment is as good or better than the old, and I define as good as being within 2%. Okay? Okay, that fudge factor is always there. If you're reading a superiority, non-inferiority trial, always be acutely aware of that fudge factor, how big that fudge factor is, and whether you think being within that fudge factor means it's the same. So, testing between groups in the equivalency approach, what I have is the new treatment, there's no difference, here's my fudge factor, and I can calculate the difference in the proportions, there it is. And then I calculate, and here's the other difference in superiority testing, a one-sided confidence bound. And so what I'm going to do is calculate a one-sided 95% confidence bound around that estimate, and it starts there, it goes up to infinity in that direction. Okay. And if you do this, two things can happen. Oh, by the way, if the 95%, the bottom end of this 95% confidence bound right there, that blue thing, does not include delta, okay, then the new treatment is as good or better than the old. Because really, if you think about what we're saying, let's see if this works again. Okay, really what you're saying is that I'm pretty sure the treatment difference is in that blue line. And if this is what I declare as no difference, if it's above that, then it must be as good or better than the old. And that is the whole idea of superiority testing. Is everybody, I see some sort of confused faces. So is everybody okay? Any questions or anything? This isn't hard. Don't make this hard. Make this easy. Okay, so the outcomes in equivalency testing, only two things can happen. You're either going to say the new treatment is as good or better than the old treatment, or you'll say there's not evidence that the new treatment is as good or better than the old treatment. Because in this case, the bottom of this confidence bound includes the delta.
Is delta the fudge factor?
Delta is the fudge factor. In carotid endarterectomy trials, your FDA has pretty much defined the same as being within 2%. And so if I'll ask you, if I was your patient and you were going to do a study on me and I had a 10% chance of dying on the table versus a 8% chance of dying on the table, would you consider that the same? Okay. Now, this delta is tightly tied to the sample size delta that you're talking about, which is defined as a clinically significant difference. We're going to come back and talk about this later. And basically, they're the exact same concept. Okay. In other words, if you're designing a superiority trial, you have to say, I'm designing this trial and I'm interested in detecting differences bigger than 2%. Okay.
How is the delta determined? Is it based on pilot data or?
Okay, so the question was, how is the delta determined? Was it based on pilot data? It's interesting. If you think about it, if you're doing a pilot study to estimate what the difference is, by definition, you don't have enough people in your study to well estimate what the difference is. If you did, you wouldn't be calling it a pilot study, you'd be calling it the study. Okay, and so the one place that you really shouldn't get the difference is from pilot data. Because you just said, I have an unreliable study that gives me crappy estimates. I think I'll use those to design the big study. And that's really, I mean, you need to say these things out loud to really get into the spirit of the junk you're doing. Okay, okay. So most of the time where the delta comes from is, we're two places, a good place and a bad place. The good place it comes from is just discussions with clinicians. How big of a difference does it take for you to change your practice? And the second, the bad place it comes from is if I say this is 3%, I can do my study with 200 people. And if I say it's 2%, I'm going to need 800 people. I think the difference is 3%. Okay? Because I can't afford to do the study at 2%. Sorry. Okay, so anyway, the advantages of the equivalence approach, I can convince me to, for a definitive statement, the new treatment is as good or better. Usually that's all a person wants. And I'll stop there and see if you have any questions, because I'm getting ready to change the topic. One-sided versus two-sided hypothesis. Okay, a one-sided assume that there's a new and old treatment, and the outcome is the proportion of people with events. Okay, and so basically a two-sided hypothesis is I assume that they're the same versus they're different. They're different can mean one is bigger than the other, or the other is bigger than the other one. A one-sided hypothesis is that the new treatment is basically as equal to or bigger than the other versus it's less than. Okay. When does logic suggest we should use a one-sided hypothesis? Basically assume that the new treatment would not be used unless it's clearly better than the old. Okay. For example, assume the new treatment is surgery, old treatment is aspirin. If unless surgery is better than this, I don't really need to prove that medicine is better than surgery. I need to prove surgery is better than medicine. This is what happened in the asymptomatic carotid atherosclerosis study in academic childhood. Basically, at the beginning of the study, there was a 2020 story about how many needless deaths there were with endarterectomy. Surely surgery would not be used unless it can be proven to be better. So a one-sided hypothesis seems logical. Why would you use a one-sided hypothesis? Well, restricting the alternative allows more power, statistical power. Basically assume that you want to see one group is 25% and be reduced to 20%. If I do this study with a two-sided hypothesis, I need to have 1,000 people. If I do it with a one-sided hypothesis, I only need to have 800 people. That's good. If I'm not interested in increased risk from surgery, then basically that's a 21% reduction in the number of people exposed. Okay, so when should you use a one-sided hypothesis? Never. Okay, you never do it. You never do it. Okay, Go find one in the literature. Go find a paper that has a one-sided hypothesis in the literature. Right there, why not? Well, basically, you're providing valuable proof. The one-sided hypothesis is used. You cannot definitively say surgery was inferior. Okay. In that case, it's difficult to discourage the procedure unless it's proven harmful. Okay. Again, going back to my dear, by the way, I only work with surgeons. I love surgeons. Don't get me wrong. My closest colleagues are all surgeons, but they will not quit doing something unless you prove it's bad. Okay. Okay. One-sided hypothesis also smells bad. If you use one, it's really hard to get your data published. Any questions? By the way, it's a lot of opinion.
But is it clear till here? This was about the hypothesis point of view, how they are going to be looked upon, and how the hypothesis is going to give you in terms of perspectives of ethics, characteristics, superiority versus equivalence point of view, one-sided versus two-sided hypothesis point of views is what we are looking out in aspects. Any questions? बेटा यहाँ पे तक कोई questions है आपको कि आपको कुछ clear है? Any questions, any doubts that you have? हाँ बेटा बोलो बेटा, आवाज नहीं आपको clear?
I did not understand what was the point of that.
No, sir.
अच्छा, ये क्यों कर रहे हैं वैसा कि I mean what's the point of statistical hypothesis वैसा बेटा? आपको क्वेश्चन क्या है?
Yeah. Like what if
हाँ बेटा, ओके मैं समझा तो मैडम, हम लोग क्या जानना चाहते हैं कि आपके पास सपोज आपने एक मेडिसिन बनाया, ठीक है, तो वो मेडिसिन इफेक्टिव है या नहीं, उसके लिए दो टाइप ऑफ थॉट प्रोसेस होगा आपके पास. ओके, एक है कि सिंपल वो इफेक्ट दिखा रहा है या नहीं दिखा रहा है. ओके, तो बेटा आपका मेडिसिन इफेक्ट दिखा रहा है नहीं दिखा रहा है, इसको डिटरमाइन करने के लिए आप क्या करोगे? ऑब्वियसली तो आपका आंसर है भाई पेशेंट पे ट्राई कर लो, ट्रीटमेंट पे ट्राई कर लो, ठीक है. तो जैसे अभी सपोज मैं हेलो हेलो,
Yes, sir.
अच्छा यू कैन हियर मी ना? फाइन बेटा. तो मैं बता रहा था, आई थिंक इट गॉट डिस्कनेक्टेड इन बिटवीन, आई वाज जस्ट टॉकिंग व्हाइल. मैं बोल रहा था बेटा कि आपने सपोज एक केस ले लिया, जैसे एक बंदे को इन्फेक्शन है, चलो अपन एग्जैक्ट इन्फेक्शन भी ले लेते हैं. सपोज मान ले जाए किसी फीमेल को यूरिनरी ट्रैक्ट इन्फेक्शन है, ठीक है. तो अब यूरिनरी ट्रैक्ट इन्फेक्शन है तो आप उसको मेडिसिन देना चाहते हो ताकि उसका ट्रीटमेंट हो सके, उसका इफेक्ट नलिफाई हो जाए. तो जब आपने उसको मेडिसिन दिया तो बेटा यहाँ पे 3-4 कंडीशंस एप्लीकेबल है. पहला पॉइंट, जब वो पर्सन को, जब ह्यूमन बीइंग को, जिस ह्यूमन बीइंग को हम लोगों ने जिस उसको पेशेंट करके आइडेंटिफाई किया जिसको यूरिनरी ट्रैक्ट इन्फेक्शन है, उसके बॉडी में उसके ही डिफेंस सिस्टम ने उसको ठीक करने की कोशिश शुरू कर ली होगी. एम आई करेक्ट बेटा? ये पहला पर्सपेक्टिव है. फाइन, डू यू एग्री गाइस? ओके, दूसरा पॉइंट है कि उसका भी अभी ऐसा थोड़ा है कि वो उसको अगर यूरिनरी ट्रैक्ट इन्फेक्शन हो गया होगा तो उसने खाना पीना बंद कर दिया या बाहर जाना बंद कर दिया, ऐसा कुछ तो नहीं है. द पर्सन वुड बी डूइंग हर ओन मेटाबॉलिक एक्टिविटीज. तो बेटा मेटाबॉलिक एक्टिविटीज में ब्रेन, बाकी के सेल्स, उन लोगों ने अपना कोऑर्डिनेशन करना शुरू कर लिया ऑफ इवेडिंग सर्टन फंक्शंस एंड ट्राइंग टू टेक ओवर सर्टन फंक्शंस, ये भी चालू कर दिया. अच्छा, सेकेंड पॉइंट हो गया. थर्ड पॉइंट, हो सकता है उसने फूड में ऐसा कुछ लिया हो ताकि उसको कुछ थोड़ा अच्छा लगे. तो ये सब आपके मेडिसिन के अलावा वाले पॉइंट्स हैं जो उसके लिए एप्लीकेबल है, ठीक है. अब उसके बाद बारी यह है कि आपने उसको अपना मेडिसिन दिया, तो आपके मेडिसिन के द्वारा उसको 3 चीजें हो सकता है. पहला पॉइंट, आपका मेडिसिन डायरेक्टली जाके वो बैक्टीरिया, वायरस या फंगस जो भी रहेगा उसको डिस्ट्रॉय कर सकता है. तो ये 100% भी हो सकता है, 50% भी हो सकता है, 25% भी हो सकता है, ठीक है। नंबर 2, जब आपका बॉडी में वो केमिकल सब्सटेंस गया तो बॉडी में आधे सेल्स ने आपके केमिकल सब्सटेंस को बाहर निकालने के काम में लगा दिया होगा और आधा आधा मेडिसिन ही आपका ट्रीटमेंट वाले में एग्जैक्ट इफेक्ट में जा रहा है। तीसरा, आपका मेडिसिन जितना अंदर गया उससे उसको उसी रेट में आपके किडनी ने बाहर निकालना शुरू कर दिया। ये 3 हो सकता है। तो ये तीनों पॉइंट हो और इसके बावजूद भी बंदे का ट्रीटमेंट हो जाए, ओके, तो आप किस बेसिस पे बोल रहे हो कि आपके मेडिसिन ने काम किया है? तो इन सब को रूल आउट करने के लिए बेटा हमको ये बताना पड़ेगा ना कि स्टैटिस्टिकल प्रोपोर्शनैलिटी मतलब कि आप इन कंपैरिजन बता रहे हो, इन कंपैरिजन टू ऑल दीज एक और पर्सन है जिसको आपका मेडिकेशन नहीं मिला और उसका यूरिनरी ट्रैक्ट इन्फेक्शन सीवियर हो गया, तो आप उसके कंपैरिजन में करना चाहते हो। तो आप ये बताना चाहते हो कि आपका इन कंपैरिजन टू अदर और नॉन टेकिंग ऑफ द मेडिसिन इफेक्टिव इसके लिए है क्योंकि ये सारे पैरामीटर्स में वो बेनिफिशियल है। तो इन सब चीज़ों को आपको अगर कैलकुलेट करना पड़ेगा तो आपको मैथमेटिक्स यूज़ करना पड़ेगा बेटा। तो बायोलॉजिकल मैथमेटिक्स जो है, बायोफील्ड में जो मैथमेटिक्स यूज़ किया जाता है वो वर्क करता है बेटा प्रोबेबिलिटी पे। प्रोबेबिलिटी मतलब चांस, ओके। तो प्रोबेबिलिटी पे वर्क करता है और इसको आप वैलिडेट करने के लिए जो प्रिंसिपल यूज करते हैं उसको बोलते हैं स्टैटिस्टिक्स। स्टैटिस्टिकल प्रिंसिपल्स और वैलिडिटी के बिना आप ये प्रूफ नहीं कर पाओगे कि वो एक्सपेरिमेंट आपका 100% इफेक्टिव है, 70% इफेक्टिव है या जितना परसेंटेज भी इफेक्टिव है। इसीलिए बेटा हम लोग स्टैटिस्टिकल एप्लीकेशंस देख रहे हैं और जब हम लोग स्टैटिस्टिकल एप्लीकेशन देख रहे हैं तो बेटा आप एक क्वेश्चन डाल रहे हो, क्वेश्चन इज कॉल्ड हाइपोथेसिस। आपका क्वेश्चन कैसा है, नल और अल्टरनेट हाइपोथेसिस में स्टेटमेंट कैसा रहेगा कि आपका मेडिसिन काम नहीं कर रहा है, ये आप पहले बोलोगे। तो जब आप बोलोगे आपका मेडिसिन काम नहीं कर रहा है, नॉर्मल एक्टिविटी की बाकी फंक्शन बाकी कंडीशन से अबाउट हो रहा है, बट आपका अपोजिट, इसका हाइपोथेसिस का अपोजिट क्या रहेगा? अगर आपका वैल्यू दिख रहा है कि नहीं आपके ड्रग के कारण वो ट्रीटमेंट इफेक्टिव हुआ, ओके, तो ये दोनों पॉसिबिलिटीज को देखने के लिए आपने हाइपोथेसिस या स्टैटिस्टिकल सिग्निफिकेंस फिगर यूज किया है। ओके बेटा, मेक सेंस? इसीलिए हम लोग बेटा स्टैटिस्टिक्स हर एक्सपेरिमेंट में यूज करते हैं। भले ही आपने लेबोरेटरी में किया हुआ एक्सपेरिमेंट हो, आप ऐसा मशीन के द्वारा करवाया हुआ एक्सपेरिमेंट हो आर्टिफिशियल इंटेलिजेंस के द्वारा, या फिर आप क्लिनिकल रिसर्च में करवाया हुआ एक्सपेरिमेंट हो, एक्सपेरिमेंट कहीं पे भी होगा, उसका वैलिडेशन ओनली एंड ओनली पॉसिबल इज विथ स्टैटिस्टिक्स, and hence statistical validation is the most important point for explanation point. Okay beta, did I answer your questions?
Yes sir. So superiority,
Okay.
हम उसमें what we are seeing basically is how superior and the drug is.
Inferior बेटा, ये कंपैरिजन है बेटा, क्योंकि हर एक बंदा यही चाहता है कि जब आपका मेडिसिन हो, आपका प्रोडक्ट हो, तो आप यही बोलोगे ना कि भाई मेरा प्रोडक्ट तो सुपीरियर है। तो सुपीरियर किसके कंटेक्स्ट में? एक स्टैंडर्ड के कंटेक्स्ट में। वो स्टैंडर्ड के कंपैरिजन में सुपीरियरिटी है या इंफीरियरिटी है, वैसा आप चेक करते हो। ओके बेटा, मुझे मालूम है थोड़ा सा कॉम्प्लेक्स है बेटा, आई नो दैट, इसीलिए मैं स्लोली जा रहा हूँ। तो ये स्टैटिस्टिकल सिग्निफिकेंस पार्ट आपको मैं इसलिए जस्ट रन थ्रू करवाया। डिटेल स्टैटिस्टिक्स बेटा लेटर पार्ट में है, विच आई विल ओनली बी टीचिंग यू, नो प्रॉब्लम, बट राइट नाउ जस्ट अंडरस्टैंडिंग पॉइंट ऑफ व्यू के लिए इंपॉर्टेंट है बेटा। क्लिनिकल रिसर्च में फेज 1 क्लिनिकल ट्रायल्स, फेज 2 क्लिनिकल ट्रायल्स, फेज 3 क्लिनिकल ट्रायल्स में कैसा कंफर्म है उसके पॉइंट ऑफ व्यू से। ओके, सो आई एम गोना शेयर अगेन बिकॉज आई नो इट गॉट डिस्कनेक्टेड अगेन। Okay guys, can you see the screen now? Can I play?
Yes sir.
Okay.
In my lectures, in case you don't get the sense of that. So we've done the hypothesis. Now how about eligibility? Who do I put into my trial? Who do I want in my study? Okay. So basically, who do you want in your randomized trial? Well, there's a great need for homogeneity of my patient population. If my patient population has divergent or weird people in it, okay, introduces noise in the system, okay? And it can really distort the outcomes of the majority. A lot of times in our study, we don't put stroke patients in our trials that are under 45 years old, because people under 45 have strokes for fundamentally different reasons than people over 45. Could be drugs play a bigger role in young people, could be congenital conditions play a bigger role. It's not that atherosclerotic process that we're trying to fight. Okay. So basically, the restriction of the population reduces noise and allows me to do my study in a smaller sample size. I don't know if y'all have ever heard of the Mr. Fit study. I won't go there. That didn't skip that. I'm running behind schedule. But anyway, having a homogeneous population makes it so I can do it with a smaller number of people. Restricting the population to a homogeneous group is good. Alternatively, at the end of my study, I want to be able to generalize my results. Okay? And basically, at the end of the study, it's important to only apply the findings to the people who are like the people in the study. Okay, if I do a study in men, I can't generalize those findings to women. Okay? So basically, it's questionable to whether or not the generalized studies can be generalized to those excluded from the study. And so basically, having a broad inclusion criteria that's welcoming to all is really a good thing. Okay? And obviously, these two things are perfectly and diametrically opposed. Okay? You want a homogeneous generalizable population is the answer to your question. Okay? So what's the correct answer? And the answer is there is no correct answer. Okay, it's a real judgment thing. And when you're reading trials, stop and think about who did they put in this study? Can this be generalized to my patients? Can I use these results? Yes, ma'am.
So when you were discussing the trial with the estrogen and the Women's Health Initiative in the beginning, they obviously had a problem when they were designing all of those observational studies, or I guess picking patients for those observational studies, or was there really nothing they could have done in the observational
Okay,
studies?
so the question is, going back to the estrogen trial, was that a matter of inclusion criteria and including the right kind of people in the study? It turns out that observational epi and in clinical trials always give the exact, precise, identical answer, always, as long as two things are true. Number one, you understand what all the risk factors are. And number two, you've measured them perfectly. Okay, the measuring them perfectly. So if I talk about socioeconomic status, how do I measure socioeconomic status? Well, by the way, the people doing the observational epi studies understood this. They tried to co-vary for socioeconomic status. They tried to fix this problem. How do we measure socioeconomic status? By far, the most common way is income and education. Okay, I have income and education. People make a lot of money. They're high socioeconomic status. I live in Talladega County. Okay, there's a racetrack over there, and there are people who make a lot of money around that racetrack. And when you meet them, you're not impressed with the underlying gestalt of their high socioeconomic status. We all know people who make a lot of money, and they're not. This sounds horrible, but it's just hard to do a discussion with them. And income is not that measure. And I mean, and the world is filled with educated derelict. I mean, we all know very educated people. And by the way, we also know very uneducated people who, are just my best friend. So we do a really crappy job of measuring socioeconomic status. By the way, if I'm offensive to anybody, accept my apology. But this is just, we do a bad job of measuring socioeconomic status. Unless we measure it perfectly, it opens the door for something called residual confounding, which means it's very much like cancer surgery, when you get some of the cancer, but not all of the cancer. I've adjusted for some of that confounding factor of socioeconomic status. I've done the best job I can, but there's still some stuff in there. And probably that's what happened to the hormone replacement, is we just didn't measure socioeconomic status well enough. There are other theories why that came out that way too, but that's what I believe. Anyway, so who do you want in your trial? Clearly a balancing act. How weird are the weird patients? You know, the results, can they be accepted for all subgroups, or is it really just one disease? Will subgroup analysis be required under any condition? You know, you just have to use good judgment. Sorry. It's difficult to depend on subgroup analysis. My favorite subgroup analysis, when my wife is very much into women's health and women's issues, and you know, your NIH has very carefully made a statement about who, including women in studies. They say you must include an adequate number of women for a valid estimate. By the way, a very carefully worded sentence. Valid means unbiased. It doesn't mean precise or good or reliable. It just means that you get the right answer on average. Okay? It doesn't mean that you can tell if there's differences between men and women. It just means that whatever you estimate on average, if you did 10,000 studies and you averaged your male-female difference in your 10,000 studies, that answer would be correct. Okay? And I'll come back to subgroup analysis in a minute too. In fact, I'll come back to it very soon. Anyway, subgroup, you know, it's common to approach special groups of people, subgroup analysis. You know, basically, in addition, you know, men and women, is this treatment equally efficacious in men and women? In addition, it's natural and important clinical questions. You know, there are subgroups in the population where the treatment was particularly efficacious, or there are places where it didn't work. You know, these are natural questions, but require careful planning. You have to really plan these things out. You know, and it sounds like I'm talking about confounding, and I thought confounding was not an issue, right? But that's not, this is effect modification, okay? Is the treatment effect, is it modified by what subgroup you're in? That's a different question than confounding. I wish I had more time to talk about that. So let me give you an example of subgroup analysis. And so basically, suppose I'm interested in and then I'll take a break after this, the relationship of diabetes and smoking to stroke among those with advanced atherosclerosis. And I use data from the ACAS study, that one that we talked about earlier. These are real data, real analysis. Okay? And it was motivated, you know, basically this is a randomized clinical trial of surgical management with carotid atherosclerosis. In that group, there were 800 people measured mathematically, and those are the people I used. Of those, 79% were normal and 21% were diabetic. And this is an example in this context of an analysis of one arm of a clinical trial. It's not really a clinical trial, but I'm just using this as an example to show you the kind of mischief and trouble you can get into. Okay. So basically, one of my heroines is Elizabeth Barrett Connor. If you don't know Elizabeth Barrett Connor, go do an email, a Medline on her, and you'll be impressed. She's a cool woman. And way back in 1984, she wrote a paper about the interaction between cigarette smoking and diabetes. And in that paper, what she noted was basically that cigarette smoking is a lot worse for you if you're diabetic. You don't want to, it's bad for you anyway. But if you have a patient who's a diabetic smoker, they're in deep trouble. Okay, and that was the motivation for this subgroup analysis that I did in ACAS. And so basically,
so guys, this is about the subgroup analysis and the details about it. Okay, so what I wanted to give you an idea was the clinical research perspective is a little bit more complex than what is assumed out, because what we normally know is, okay, clinical research means just to go ahead, know that phase 1 ट्रायल में कितने बंदे होते हैं, phase 2 में कितने बंदे होते हैं, क्या स्टडीज चाहिए, मतलब मेडिसिन देने का रहेगा, मेडिसिन अगर फर्क पड़ा तो मेडिसिन गुड फॉर ट्रीटमेंट एंड दैट्स लाइक दैट. बट इतना सिंपल भी नहीं होता है. इट्स अ लिटिल बिट कॉम्प्लेक्स स्टफ एज यू कैन सी राइट नाउ. ठीक है बेटा. तो ये कॉम्प्लेक्स स्टफ जल्दी से लोग समझ नहीं पाते हैं. दैट्स द पॉइंट विच वी आई वांटेड टू मेक यू अंडरस्टैंड. तो बेटा आपके ईज ऑफ अंडरस्टैंडिंग के लिए, आफ्टर हैविंग लुक्ड एट द हाइपोथेसिस सबग्रुप एनालिसिस तक का डिटेल्स, I am taking to a quick one where you can just see the gist of it. Okay.
Thank you.
Thank you very much.
Wait a sec, guys. I'll just,
So one announcement before.
Just give me a second, guys. I'll just load it up for you.
What is a clinical trial?
Guys, आपको स्क्रीन दिख रहा है बेटा? Please respond. Hello. Can you see? Okay बेटा. तो बेटा ये मैं आपको 5 मिनट में वो जो कॉम्प्लेक्स स्टडी के बजाय मैं एक जिस्ट फॉर्मेट में दिखा देता हूँ. तो you'll get an idea about the clinical trial perspective. इसका डिटेल वर्जन इज व्हाट आई कैन टेक, बट व्हाट इज द थिंग इज आपको मेरे को सिर्फ हाइपोथेसिस तक का दिखाने का था, हाइपोथेसिस ट्रायल तक का, क्योंकि दैट इज द पार्ट विच इज इम्पोर्टेंट, क्योंकि वो आपके बेटा स्टैटिस्टिकल प्लानिंग में आता है एंड दैट इज पार्ट ऑफ योर रिक्वायरमेंट, क्योंकि आपको उतना जानना ज़रूरी है. उसके आगे भी मोर डिटेल्स हैं जो आपको तब तक जानने की जरूरत नहीं जब तक आप कोई क्लिनिकल रिसर्च कोर्स ना कर ले, ऐसा कुछ हाई एंड ना करे या क्लिनिकल रिसर्च इंडस्ट्री ज्वाइन ना हो जाए. सो आई एम जस्ट गिविंग यू एन आउटलाइन ऑफ इट, क्लिनिकल ट्रायल इन एन आउटलाइन.
Clinical trials are research studies to test potential new medicines or new uses for old medicines. Healthy volunteers or patients with the illness that is being studied participate in the trial. The aim is to gather enough evidence to see if the medicine works and if it is safe. The data from clinical trials are sent to regulatory authorities who review it and decide if the medicine can be brought to market. Clinical trials rely on people volunteering to participate. It is only through clinical trials that science can make advances in treating diseases. Clinical trials go through a long review process before they can begin. Each trial starts with a protocol designed to answer specific research questions and to safeguard the health of the participants. It describes who is eligible to participate. It lists the tests, procedures, and medications involved and what schedules each of these will follow. It tells how long the study will be, details what information will be gathered, and how it will be analyzed. Protocols are carefully reviewed by the scientists, physicians, and the investigators who will be conducting the trial. Every study is led by a principal investigator, an experienced doctor who agrees to follow the protocol precisely. Clinical trials also involve a research team that may include nurses, social workers, and other healthcare professionals. The final protocol has to be submitted to regulatory authorities, such as the FDA and ethics committees or institutional review boards, IRBs, for approval. Clinical trials in the U.S. are reviewed, approved, and monitored by an IRB. An IRB is an independent committee not associated with the company conducting the study. The IRB reviews the study to be sure it is ethical and that the rights of the participants are protected. The government has enacted laws to make sure clinical trials are conducted correctly. These laws also ensure that all investigators and their staff are fully qualified, licensed, trained, and certified to conduct the study. Clinical trials can only begin after the regulatory authorities and the ethics committees or IRBs have given approval. There are many different types of trials, and their design will change for the stage of research and to meet different objectives. Generally, for new medicines, trials are done in phases, with the first studies being small and gathering information to better design the next trials. Phase 1 trials test a potential new medicine for the first time in a small group of people. Phase 1 trials are to gather the first round of information about the safety and side effects of the investigational medicine or device in healthy volunteers or patients. Phase 2 trials give the drug or treatment to a larger number of people, usually with the disease being studied, to figure out the best dose and begin to measure how well it works. The larger number of people means more information on the safety of the medicine. Phase 3 trials are conducted in an even larger number of patients, often thousands. The main aim of these trials is to gather clear evidence of the efficacy, or how well the drug works, and safety of the investigational treatment. When an investigational medicine is studied, it is not usually known if it will be helpful, harmful, or any different than already available treatments. The new medicine may turn out to be less effective or less safe. A new treatment is often compared in one study to the best existing treatment, so researchers are able to better understand and interpret the results. Sometimes a trial will compare the investigational medicine to a placebo or sugar pill. This is not done if it's a serious illness where patients would be at risk from their disease because they are not receiving any active treatment. Cancer studies often compare patients receiving the best currently available treatment to those receiving that same treatment plus the study medicine. In studies like these, patients are randomly assigned to one group or the other, like flipping a coin. In many studies, neither the patient nor their doctor will know which group the patient is in. This blinding helps researchers get more accurate and unbiased information as the results are not affected by what the doctor or patient thinks. Someone, usually a pharmacist, has the key and can tell which patients are on the study drug if it's needed to know for safety purposes. There are risks and benefits in every trial, and they are not the same for everyone. Because participants are different, they will each experience the risks or benefits differently. One person may have a certain side effect, while the next person may not. The risks and benefits of a trial are hard to predict. Generally, researchers do not start a trial until they are hopeful the potential benefits outweigh potential risks. Even though efforts are made to control risks, it is not possible to avoid all because the risks of the investigational medicine are not known. It is important the patient makes the decision to participate only after they have a full understanding of the process and possible risks. If someone is thinking about participating in a clinical trial, they will have a detailed discussion with the primary investigator or study doctor on the study about the trial and will review an informed consent form. The ICF reveals what a patient needs to know about the study. If they decide to participate, they will need to sign the consent form along with the study doctor. A patient can always change their mind after signing, even if treatment has started. The ICF is the document to help make an informed decision about participation in a clinical trial. It is only with the help of volunteers willing to be in a clinical trial that we can learn more about a disease and find better ways to treat it.
Okay, guys, this would have been very easy for you now for understanding purposes, beta. Hello?
Yes, sir.
हाँ, ये बेटा जिस्ट में इजी पढ़ा होगा आपको, क्योंकि आपने जैसे क्वेश्चन पूछा था ना बेटा कि रैंडमाइजेशन क्या है, वो भी इसमें कवर अप हो गया आपका। इन जनरल जो डिस्क्रिप्शन है क्लिनिकल रिसर्च वाला, यानी क्लिनिकल ट्रायल्स का, व्हाट इज क्लिनिकल ट्रायल, व्हाट आर द फेजेस ऑफ इट, वो इसमें कवर अप हो जाता है बेटा। ओके, so is it clear beta? This is absolutely clear. So then what I can do is in the next lecture we'll be completing the section of the NC part जो अपना है वो अपन complete कर लेंगे, where we will be completing 7 and 8 and the clinical trials का कुछ fundamental details. Meanwhile मैं बेटा आपको आप लोगों को clinical trials का जो information है in terms of what is the phases of clinical trials, इसका article form या PDF form में भेजने वाला हूँ, so you can just go through it. But बेटा उसका elaborative details is a question which is dependent on you. अगर आपको elaborative details पूरा details में study करना है तो I can take up the explanation, or otherwise it can be skipped through because वो बेटा आपके लिए directly नहीं है except for basic fundamentals of clinical trials. Okay बेटा? Okay guys, thank you so much beta, that I believe I have been able to dispense out a lot of information regarding clinical trials, and you got an idea about how the statistics works in clinical trials in that perspective. So this is what I wanted to cover today. So I'll see you soon in the next lecture where we can continue ahead with the remaining part of it. We'll be touching base upon 1.4 ka 7 and 8, which is compliance to ethical guidelines and dealing with ethical guidelines, and the clinical trials का fundamental basics का कुछ part और. Okay बेटा? Is it fine guys?
Yes, sir. Yes, sir.
Okay बेटा. Yes. Thank you so much for attending it बेटा. Thank you so much for attending the lecture. I enjoyed teaching you all and giving you the perspective of what exactly is part of clinical trials. So see you soon in the next lecture. Till that time I meet you. I wish you a goodbye and good day. Thank you so much.
Sir.
हाँ बेटा बोलो.
Sir. Sir,
हाँ बोलो।
these videos, can they be found on YouTube, or is it like not,
बेटा
or is not
आप जो
available freely?
ये जो वीडियोस है ना बेटा, लॉन्ग टर्म वीडियोस जो है, छोटे वीडियोस आपके अवेलेबल है, जो 3 to 4 मिनट्स वाला है, आपको मिल सकता है। ये लॉन्ग वीडियोस जैसे मैंने आपको बताया ना, जो इन डेप्थ स्टडीज का होता है ना बेटा, ये वीडियोस आपको यूट्यूब पे नहीं मिलेगा बेटा, डायरेक्टली नहीं मिलेगा। यू हैव टू आस्क द रिस्पेक्टिव पर्सन फ्रॉम हु इज प्रेजेंटिंग इट आउट। मेरे पास ये इसके लिए है क्योंकि आई हैड आस्क्ड द कॉन्फ्रेंस साइट्स एंड दोज गाइज हु आर प्रेजेंटेड आउट तो सेंड अक्रॉस टू मी बेटा, बाकी वो डायरेक्टली अवेलेबल नहीं कराते बेटा। ओके,
Okay, sir.
छोटे वीडियोस मिल जाएंगे आपको बेटा। और आपको कुछ अगर चाहिए रहेगा एक्स्ट्रा वीडियोस पॉइंट ऑफ व्यू में चाहिए तो आई कैन सेंड अक्रॉस ऑल्सो। छोटे वीडियोस मिल जाते हैं, बिग वंस नहीं मिलेंगे बेटा आपको। ओके, बिग वंस डायरेक्टली
Okay,
नहीं
sir.
मिलते। ओके बेटा, थैंक यू सो मच बेटा। सो आई सेंड अक्रॉस द